Okay, so we were talking about the life cycles of RNA viruses, and I got one left that I wanted to finish. So if you remember, we'd gone into the principles of what's different between putting message into the cell, those are the positive sense RNA viruses, from putting complementary negative sense RNA into the cell. And we discussed the non-segmented negative strand viruses, which were rabies, or that's rhabdovirus family, the paramyxovirus family. And then we discussed influenza virus, an unusual RNA virus in that it goes to the nucleus. And it's still negative sense, but this one was segmented. And we'll come today to talk about some of the significance of that segmented genome. And does this audio sound okay, or is it rather echoey? It's okay? It's quiet. Okay. Tell my husband that I'm quiet, he'll never believe it. Okay, does that sound a bit better? Well, tell me if it doesn't, somebody. Okay. Anyway, so we talked about flu, and flu goes to the nucleus, and that's really unusual for the RNA viruses. So again, if you remember the exceptions, in this course, the only exception that goes to the nucleus of the RNA viruses that we're going to talk about is influenza. So, and because it goes to the nucleus, it will be the only one that has splicing of the ones we talk about. Okay, so I want to get started on the last family. And the last family that I want to talk about of RNA viruses in this lecture are the reovirus family. Reo originally meant respiric, enteric, orphan viruses. An orphan is a terrible name to give to a virus. It means you don't know what disease it causes, if any. But the problem is a lot of these orphans turn out not to be orphans, so it's a terrible nomenclature. Anyway, that was what the Reo stood for. The major one that we're interested here, you'll be hearing about from Dr. Albrecht in the following lecture, is rotavirus, because that's a major problem with diarrhea, particularly in infants. So it's a major significant cause of death and morbidity worldwide. In this country, fortunately, you can deal with the diarrhea pretty well, and we have vaccination, so it's going down in significance in this country, but where those kinds of approaches are not available, it's a major problem. So it's still a problem here. It's got a double-stranded RNA genome, and if you remember, I said that as far as the ribosome is concerned, double-stranded doesn't function as message. They can't unwind it to find the message strand. So this is going to behave as if it was a negative strand virus. In other words, it's going to have to make its own message because the ribosomes cannot read this stuff as message. It's segmented, which will also have significance later on. And it has a very solid capsid. It has a sort of double nuclear capsid structure, and it's really very tough. It has no envelope, so it's not easy to inactivate, which is why it's so much of a problem in kids, because kids' sense of hygiene is not brilliant and it's not easy to inactivate, and so you put the two together and you've got major problems. In addition to which, it's transmitted fecal orally, and when you've got dirty diapers that are full of millions and billions and trillions and whatever numbers of these virus particles, toss them somewhere and it spreads. So this is a very tough little virus, very difficult to control. And because, as I say, it's behaving as a negative, in a similar way to the negative sense RNAs, it has to take in a polymerase. All of these RNA viruses have to code for a polymerase. We don't provide it. But the ones that don't function immediately as message have to take the polymerase into the cell to make that message. So it's going to do that. This just explains, as I say, the major subgroup of this that we're going to be concerned with are the rotaviruses. Rota means wheel, as in rotary. And if you look here, and you probably can't see it from where you're sitting, but it does look like they got the outside of the virus particle has things that look like spokes of a wheel, and hence the name rotavirus. As I say, the most important human pathogens are the rotaviruses that Dr. Albrecht will be talking about, but I will briefly mention a virus that is transmitted by ticks later on, which is called Colorado tick fever, and it's a minor thing. So they attach to receptors on the cell. This time they're going to come in orally, which gets to be important, they survive the stomach acid really well. They're very tough little things. And then they get into the gut, and they've got the same old problem. If you're really tough and you're not going to take your armour off, you can't do much. And we take their armour off for them because we've got proteases in our GI tract. So we actually chew off that outside shell that saw them all the way through the stomach and protected them. And so if this is one of these image reconstructions of the virus particle, and what happens is we chew off this outer layer, so we chew down on it, and as we chew down on it, the pentons, the vertices, were sort of buried inside, and they pop out, they kind of flower. And so you get these 12 little flowers at the vertices, and those are the attachment proteins. So the attachment proteins are now released from being protected inside that outer shell. They pop out, they flower, and they can now bind to the receptors on your GI tract cells. Okay, so as I said, it's proteases in the GI tract that actually release those receptors so that they can attach. The proteins are at the vertices, those 12 pentons. And they cross the membrane either directly or via endosomes. I mean, it is really difficult to look how a virus gets inside a cell in your GI tract. So we assume this from various studies, but I'm not going to quiz you on does it go across the membrane or does it go via endosomes. It probably uses either. Okay, so this is going to be, I hope, a pretty simple life cycle to go through. Here's the parental virus, which will lose its outer shell in your GI tract or anyone's GI tract. And then the nuclear capsid somehow gets into the cell. And we don't know how it gets into the cell, but it gets into the cell as an icosahedral nuclear capsid. So when you look in the cytoplasm, you have a somewhat stripped down version of an icosahedral nuclear capsid, but it's a capsid all the same. So the double-stranded RNA is kept inside the virus particle. And that, if you notice when we complete morphogenesis, double-stranded RNA is always inside the virus particle for this. And we'll see tomorrow when we come to discuss interferon why that is. But the quick answer is double-stranded RNA tells the cell that something bad is going on, and it routes an antiviral response. So if you keep your double-stranded RNA inside your nuclear capsid, you protect yourself from triggering the antiviral response. So there is a logic for why these viruses try and don't have too much double-stranded nucleic acid around, and this is one way. The cell doesn't know it's in here. So the polymerase actually works inside the nuclear capsid, just like we had with the negative sense RNA viruses, where the RNA didn't have to uncoat, and the nuclear capsid protein remained associated with it and provided protection. We have the same thing here. It doesn't have to uncoat, and in fact, it's probably better if it doesn't uncoat. And so the polymerase is inside this nuclear capsid because it's got to make message. It makes message, and you know I said those pentons sort of flowered at the vertices when that protein was removed. What actually happens is those flowers become like chimneys. They've actually got holes in the middle. Now, obviously, they don't have holes in the middle when they're in the gut, but by the time they get into the cell, this has undergone conformational changes, and there are now holes in the middle down each of those pentons. And so the messenger RNA that's made comes out through those chimneys, and you actually get RNA being extruded out through all your pentons. And, of course, nucleotides can get in. It's amply big enough for the nucleotides to get in. So you can get all the substrates in here. And then the RNA comes out through these pentons. And actually, at one stage, I mean, it may not be your idea of a great Christmas card, but virologists had these Christmas cards which have pictures of this virus particle with like stars with all the little RNAs coming out of the various pentons. Okay, well, forget about it. Anyway, they make the message. It's released into the cytoplasm. It's translated into proteins. The nucleus plays no role in this. And then what happens is some of those, there's no early or late stage here, but some of those nuclear capsid proteins assemble around messages. So now what you've got is you've got a message inside these particles. And obviously, once the messages get surrounded by a nuclear capsid, they're no longer going to be translated into proteins. But the proteins keep on being made from the RNAs that haven't been encapsulated, and gradually you get more and more proteins accumulating. And then it's like it all of a sudden says, gee, I'm supposed to be a double-stranded RNA virus, and it copies that message into the complementary strand and makes all these double-stranded. So now these double-stranded RNAs are ready to complete the morphogenesis. More nuclear capsid proteins are added, and you finally get a complete virus particle that gets out of the cell by lysis. So are there any comments on that? So again, keep it double-stranded inside the nuclear capsid here, release single-stranded messenger RNA, encapsulate single-stranded messenger RNA, and then make it double-stranded, so you keep it hidden from the cell. I put a coverall thing, completion of morphogenesis here. And there's a very odd thing that occurs with the rotaviruses, and we don't really understand what it means. I'm just going to mention it just in case you see any comments on it, and maybe soon, later on, we'll discover what's important about this. But for some reason, these rotaviruses go into the endoplasmic reticulum and get an envelope. And then they come back out of it. That envelope fuses with the endoplasmic reticulum, and they go back into the cytoplasm. So I'm just telling you, I'm not going to examine you on that, but I just want you to be aware of that there is this funny envelope stage. And it may be a target, but we don't know. So they're still released by lysis. So they just sort of kind of pop in, say hello, and pop back out into the cytoplasm. So it's very transient. And as far as we know, the other members of the reovirus don't necessarily do this, but the rotaviruses do. That's all I want to say about rotaviruses. Are there any questions?